The overall goal of this procedure is to yield a passivated quartz slide surface of superior quality. This is accomplished by first cleaning drilled quartz slides and cover slips with water and acetone. Then, the slide is etched with potassium hydroxide and the piranha solution to enhance the quality of the surface. Next, the slides and cover slips are functionalized with amine groups, followed by 2 rounds of surface passivation using a polymer to make the glass surface inert. The final step is to assemble the microfluidic chamber from the slide and cover slip with the polyethylene glycol coated or pegylated surfaces facing each other. Ultimately, a new strategy of treating the surface with PEG molecules over 2 rounds remarkably improves the quality of passivation as observed by single-molecule fluorescence microscopy.
The main advantage of this technique over the existing techniques such as polymer coating after potassium hydroxide etching is this technique guarantees superior quality.
People new to this method will struggle since multiple surface treatments have to be performed.
To begin, drill the quartz slides as detailed in the text protocol. Clean the slides by placing the slides in a glass staining jar. Typically, 5 to 15 slides can be placed in a single jar. Then, rinse the slides with milk.